Okay, so we're doing the second part or the second half of chapter 3, Bio131. This is the second half, and we are talking about the chromosomes. So humans have 46 chromosomes. Do we have any idea about the chromosomes? What are the chromosomes? Yes?
There's two different types of chromosomes, X and Y.
X and Y. These are some types of it. It's called the sex chromosomes, but it is type of the chromosomes, yes.
You have the genetic information.
Carry the genetic information, inheritance, yes.
46
6 chromosomes that he mentioned and you mentioned, it's part of it. There are 46
Two are the same,
and the other ones are 6 chromosomes.
Does it contain the DNA?
It contains DNA, yes. It's DNA wrapped around proteins, basically. So if you look at this picture right here, do you remember when we talked about the nucleus?
Yes.
What is the major difference between the prokaryotes and eukaryotes?
Cell wall.
Pro is units.
Pro,
Is before, is it like?
so does it have a nucleus?
No.
No, it's not have a nucleus. Does it have genetic material, DNA?
No.
The prokaryotes?
Yeah.
Yes. If it doesn't have it, it wouldn't survive. It does have genetic material, it does have DNA, it does have chromosomes, but it's not surrounded by a membrane. You do not call it a nucleus. It is not a nucleus. Because where is the nucleus? Like, you have to have a membrane, right? If you don't have walls, you don't call it a house. If you don't have a wall on the nucleus, you don't call it a nucleus. It's genetic material, okay. DNA, chromosomes, that's fine. But you cannot call it a nucleus. What's the other thing that's different?
It's in a plant and animals.
Cell wall is in prokaryotes, but in eukaryotes, some of them have cell wall. Like plants, for example, they have cell wall. What's in a pro only?
Bacteria.
The grains.
Capsule. Do we have capsule in eukaryotes?
Yeah. No.
No capsules. No capsules. What else? Eukaryotes have what else? Huh? Chromoplast. This is in the plants, but I'm talking about eukaryotes in general. How about mitochondria? Eukaryotes have mitochondria,
Yeah.
right? What else?
They don't have plasmids.
Eukaryotes have membranous organelles. Prokaryotes do not have membranous organisms. You remember this? Okay. So these are the major differences. Eukaryotes have the following. That's not in the prokaryotes. Number 1, eukaryotes have a nucleus with a membrane, nuclear membrane. Okay? This is 1. Number 2, eukaryotes, all of them have mitochondria, plus or minus other things, but they have mitochondria. Number 3, eukaryotes have membranous organelles.
What is it?
Membranous organelles. Organelles that are covered by membrane, not just a nucleus, other things that are covered by membrane.
What's it called?
Membranous.
Membrane.
Like membrane, us, OUS. Membranous organelles. Okay? So if you look at this picture, we're talking about the organelles, the membranous organelles that I talked about, which is organelles that are surrounded by membrane here. And what falls under the category of eukaryotes? Can you give me examples of eukaryotes?
They're multicellular or single cellular.
They can be multicellular or unicellular or singular cellular, but give me examples. Like what?
Plants.
Plants, that's one. Animals, that's 2. Huh?
Fungi.
Fungi, yes. What's the last one? Protist. And a protist is the only example of unicellular. It's unicellular eukaryote. Others are not unicellular, right?
So the 4 categories are animal, plant, fungi, and last one is
Protist.
protist.
Protist. Okay? Okay, so here we're talking about the plants. How did I know that this plants? Can you tell me how did I know? I look at this and I say plants.
Cell wall.
Nucleus.
The cell wall.
plants,
Cell
sorry.
wall.
Cell
Wall.
wall. What is this?
Membrane.
This is a membrane. That's not a difference. That's not a difference, right? Everything have a membrane. But what's outside of the membrane?
The wall.
Cell wall. So it's plant. That's one. What else? What else should you see here?
Chloroplasts.
If to tell, it's chromoplast. That's two. What's the third one? What do you see here? That's not here. Vacuole. Yes, vacuole. You have a vacuole in the plants, not in anything else, not in animal cells.
The vacuole.
Vacuole. Yes, a vacuole is like a space, a bag, a big bag in which we store different things that's extra. Extra water, extra food, nutritions, anything that you have extras, just keep it in that big vacuum. Okay, but look inside here, you'll see a nucleus here and a nucleus here. An actual nucleus that's surrounded by a membrane, right? And what do you call this?
Nucleolus.
Nucleolus, which is the nucleus of the nucleus. We'll talk about the different functions, but these are the basics. So nucleus contain the DNA stored in the form of chromosomes. So if you look at both of those, can you tell me what is this? Vacuole. This is a vacuole. You can have multiple vacuoles, but you have to have one big, at least. Okay, so if you look at this, this is the nucleus. This is the nucleus, and the nucleus contains the DNA stored in chromosomes. Okay, we are going to go like from small to big, so do not confuse those with each other. Do not confuse DNA with the chromosome. Okay, DNA, chromatin, chromosome. It goes like this. We will talk about the details. So if we focus more on the nucleus, you will notice the following. The nucleus is surrounded by a double membrane. The membrane that's double layer. That's one. We call it the nuclear envelope, but it's a double membrane. You see these pores? See the pores? It have very small openings, pores,
Yeah.
to allow things to go in and out, basically out. Things like what? Things that are very small. DNA is big. It can't go through this. RNA is tiny, small. It can't go through it. So the pores are mainly for RNA because RNA is
Small.
very small. Okay? The chromosomes are filling all this space right here. Okay? In the cavity or inside the nucleus. And this is the nucleolus at the center of the nucleus itself. So this is the basic structure of the nucleus. Okay? So DNA, do you have an idea about the DNA? Do you know the DNA? DNA is it a double strand or single strand? Double strand, right? It's a double strand of nitrogenous bases, two strands, right? This is the DNA. If you have this DNA and wrap it around protein, you call that chromatin. We know where this chromatin came from, the word? Chromosome and protein. Chromatin. Merge them. Chromosome, protein. Merge, chromatin. Right? So chromatin is DNA. You roll it around the protein. Now you call it chromatin. If you get that chromatin and wrap it together more and make it more condensed, that will make the chromosomes. Did you get the idea? DNA is two strands. What do you call it when you wrap it around protein? Not chromosome. Chromatin, right? Which is chromosome, protein, merge. Okay? DNA around protein, you call it chromatin. If you put that chromatin, you wrap it together and make it more condensed. This is what we call chromosomes. Nucleolus is like the nucleus of the nucleus. It's at the center of the nucleus. And the importance of each one of these organelles is very important to know. Why do you have nucleolus? It's to make ribosomal RNA. It's one of the RNAs. Exactly. You have to remember this. This is the basic function. Why do we have it? Right? We know that the nucleus is the manager of the cell, right? And giving order to every other organelle. Okay, so this is inside of which, and the importance is to make ribosomal RNA. This ribosomal RNA can go out. Is it an RNA? RNA is small enough, whatever the type is, right? We have different types of RNA. All of them are tiny. RNA is a small molecule. Can it pass through the nuclear pores? Yes, it can. And it go to the cytoplasm where it make ribosomes. And what's the function of the ribosomes that we talked about before? Protein. Yes, protein. This is the factory of protein. This is where we make the protein. Yes. So plasma membrane. We talked about the role of the plasma membrane before, right? Plasma membrane is mainly to regulate what goes in, what goes out, right? It also to identify what's happening outside. It's telling you what's happening outside, right? Nucleus is the manager. It have the orders for everything because it have the chromosomes. Chromosomes are DNA, and DNA is the basic or the original menu to do anything. Okay? Have the original instructions to do everything. Then what's the function of ribosomes? Make proteins. What's the function of the mitochondria?
Power the cell.
Power the cell, which is energy, right? Okay. So how do we make the RNA from DNA? We get the DNA in the nucleus and you make copy of it. Can you tell me why are you making a copy? DNA is the original recipe, the original instructions to make every single protein in the cell. The question is, why do we have to make a copy of DNA, which is RNA, and why don't we just use the original copy, which is DNA? Because which one is small? Yes, it's too big. Which one is small?
RNA.
RNA. So the thing is, why can't we use the original copy? Because it's too big. It cannot leave the nucleus. Where do we make the protein? In the ribosomes, isn't it? Where is the ribosomes? Is it in the nucleus or in the cytoplasm?
Cytoplasm.
In the cytoplasm. Ribosomes, I know that we make it in the nucleus, but it leaves. Where is the final location? It's in the cytoplasm, right? And where do you make the proteins? In the ribosomes in the cytoplasm. So if I have the instructions in the room and I need to make it in the factory, how can I get the instructions out of the room if it is too big? I have to make a copy and then send that copy to the factory, which is ribosomes. Make sense? So you have to make a copy, and the copy is small enough to go through the pores and go outside to the cytoplasm, go to the ribosome. Ribosome will read the copy and make proteins. So DNA, a copy of the DNA is called RNA, and RNA will go to the ribosome through which you make proteins. And protein is needed for everything in the cells. It's needed for a lot of things. The basic structure for a lot of things. So you have to make protein anyway. These three organelles are playing a role in this process somehow. Ribosomes, these are the factories that actually make the proteins, isn't it? There are two types of ribosomes. Can you tell me what are the two types? This is the endoplasmic reticulum, rough or smooth.
Yeah.
Which one have ribosomes on the surface?
The rough.
The rough.
Yeah.
And which one does not have ribosomes on the surface? The smooth. So where is the ribosomes? The two types. One type is attached on the surface
Of the rough.
of the rough. How about the other type?
It's not attached to it.
It's what?
It's not attached to it.
So what do you call it? Free. Free. The other ones are free in the cytoplasm. Not attached to anything. Free. So we have two types of ribosomes. Attached to the rough ER or the rough endoplasmic reticulum, and the other one is free. Okay? Why is that? They do different functions. I'll talk about it. The free have a function. Both of them make proteins. Both of them make protein. So why do we have two types? Why don't you just make it one type? Because the one that's here, that's attached here, when you make the proteins, it's going to flow like this within the rough endoplasmic reticulum, and it will go to the Golgi apparatus. The Golgi apparatus is going to do, what do we call the Golgi apparatus? The finishing unit. The finishing units. After you're done with the protein in the endoplasmic reticulum, do you use it? Is it ready to use? No, it is not. It's not ready to use. You send it to the finishing unit, which is the Golgi apparatus, and the Golgi apparatus will take it, modify, finish it, package it, make it ready to export. So this is the first type of protein that we exported. Look at these. Do you know what is these? What is this blue here? What? Vesicles. Yes. So the protein enter inside the Golgi apparatus. It does the finishing, it does packaging, finishing and packaging, and you pack it within vesicles. See how the vesicles attach to the membrane and goes out? So this is for export. Like car factories, do they make some cars for exporting and some cars to utilize it here, to use it in the US, right? This is the same thing. The ones that are going to be exported are the ones that come from the rough. Why? Because it go to the rough endoplasmic reticulum, and then it go to the Golgi apparatus, and then you put it in the form of vesicles, and then you export it, meaning to push it out. Can you tell me whether this process of crossing the membrane by vesicle like this, there's a name for it if you study. If you're studying, what is it called? You see how the vesicle is attaching itself to the membrane, releasing out? There is a name for this. Do you remember when we talked about the membrane, there are different ways to cross the membrane? Diffusion, active transport. Do you remember this? So which one is this? When you fuse yourself like this with the membrane? No, there is a name for it. Exocytosis. Yes. How about the opposite? If something that's big and fuse itself in the membrane, cutting the membrane, and then moves in. Endocytosis.
Can you repeat them again? Sorry.
Exocytosis is when you fuse with the membrane and export things out. Endocytosis is when you fuse with the membrane, cut the membrane, and move things in. Endo in, exo out. So did you notice that the proteins play a role? The rough ER or the plasmic reticulum play a role, taking it from there and send it to the Golgi apparatus, and Golgi apparatus have a rule. So all three combined will make the protein for export. Yes?
So the endoplasmic reticulum is basically a pathway?
Kind of a pathway. It play another role, but not our issue. Kind of like a network that take it from here to the Golgi apparatus. So we said that we make a copy of the DNA, and you call that copy what?
RNA.
RNA. This process is called transcription. Transcription, translation, copying. Transcription is copying. When you transcribe something, mean you make copy out of it. So when you make RNA from DNA, you call that transcription. So after transcription, you're making RNA, right? This RNA, can RNA leave the nucleus through the pores? Yes, because it's small enough, right? So it's going to leave and go all the way to what? To the
Endoplasmic reticulum.
more specific. Where does it go? What does it have? Instructions to make what? Protein. So where does it go to?
Golgi apparatus.
The ribosomes. Isn't the ribosomes are the factory of the protein? Where do you take the instructions? To the factory.
Oh.
Right? The factory will take it. These are the instructions. Okay, I will make protein. Right? Anything else is after you make the protein already. Right? And the plasmic reticulum, it takes the protein. It's protein already. It take it to the Golgi apparatus. Golgi apparatus take it. It's protein already. You're doing finishing and stocking and stuff, but still it's a protein. Who made the protein in the first place? Ribosomes. So it leaves the nucleus and go to the ribosomes. Ribosomes are going to read it and translate the code in the form of protein. We call that translation. So we start with transcription and then goes to translation. Clear? Transcription, you make a copy of DNA, which is called RNA. Translation means this RNA, when it goes to the ribosomes will read, translate, make protein. We call that translation. So if you see here, DNA, you make a copy, you call it RNA. RNA is small enough, it leaves, go to the ribosomes, and the ribosomes are going to make protein, and protein goes to the Golgi apparatus, packaging it, put it in the form of vesicle. Now it is ready to use or to export. So ribosomes is where we make the proteins. It can be free, it can be attached. Is that clear? If it's attached to what?
Yeah, endoplasmic.
Making it what? Rough. Look at this. These are the two subunits. Look at how it's 2 units. The purple. Can we see the purple? It's 2 units. This is the ribosome. And do you see the red in the middle? Can we see it?
Yeah.
There's 1 unit here and 1 unit here and red in the middle. This is how they read and translate. So the RNA goes like this in between the 2 units in order for the 2 units to read it and translate it. So the question will be, we know that the rough ER, or the rough endoplasmic reticulum, is the one that have what on the surface? Ribosomes. The one that does not have ribosomes on the surface, you call it smooth. It has another function. It's where you make lipids, like steroids, lipids. So can you tell me the function of rough endoplasmic reticulum?
It's to
Rough.
ribosome attached to it. And it goes to
So it's helping in making proteins. The ribosomes on its surface make proteins. So it's helping to make protein. How about the smooth? Lipids, like steroids. Golgi apparatus, it's the finishing, sorting station that finish and sort and package the proteins in order to be able to export. You have to put it in the form of what first in order to export? You have to package it in the form of what? Vesicle. Yes, you have to package it in the form of vesicle. If you have a TV, don't you put it in a box, or you just export it as it is? You will break it down, right? No, you put it in a vesicle first. You package it in a vesicle, and this vesicle is going to export out. The next organelle that is in the eukaryotes is called the lysosomes, and I gave you an idea about all of these organelles in the very beginning. If you remember, I went through it briefly, but the lysosomes, this word consists of 2 parts. What are the 2 parts? Lyso and somes. What's lyso? Dissolve. And what's some?
body.
So this is a body that dissolve, the dissolving body, or the bodies that dissolve. Dissolve what? Anything that you don't need. Shredder. What kind of paper do you shred? Anything. Any paper that you don't need, right? So this is your shredder. You send anything that's not needed, you go to that shredder and you shred it. You break it down. You dissolve it. Examples. If you have organism enter your cell, do you need it? Do you need the organisms? Like bacteria or something entered your cell, virus entered your cell. Do you need it to stay? Of course not. You need to shred it, dissolve it, send it to the lysosomes, and it will dissolve it. How about toxins? There are toxins somehow entered your cells. Do you need it there? Of course not. Anything that's not needed, you send it to this disposal, and it will shred it, it will dissolve it and get rid of it. Medication, extra medication, get rid of it. How about even the old organelles? Like you have an old mitochondria that stopped working already. It's not useful anymore. It's useless. Do you still need it? No. Send it to the shredder, send it to the lysosomes, they will break it down. So like this example here, this is the mitochondria. So you send it to the lysosome will take it, dissolve it, and then you can recycle it if it is usable, or get rid of it if it's not usable. So the question is, what happens if you don't have ribosomes? You cannot make proteins, right? What will happen if you do not have mitochondria?
The cell wouldn't work function right.
Why? Because what's missing here? Energy. You cannot work without energy, right? Okay. So mitochondria is located in any eukaryote. Any eukaryote have mitochondria, okay? Plus or minus chromoplast in plants and some algae. Algae is not really a plant. Do you know what's algae? Did you see it in the surface of the water or inside any water, that green stuff? So it's green because it also have chromoplast. Algae is not considered plant, but it does have chromoplast, and that's why you see it green. Did you see it? It's green. Why is it green in color? Because it have chromoplast. So what are the two eukaryotes that have chromoplast? Plants and some algae. The green algae only. Do we have other types of algae? Yes, but it's not our issue. There is a red algae and other types. I'm talking about the green algae. Some of the algae, okay? They have the chromoplast. So the plants have chromoplast. Do they have mitochondria? Yes. Any eukaryotes have mitochondria. Any eukaryotes have a nucleus. These are basics now, okay? Are we following? Any eukaryote have a nucleus with a membrane. Any eukaryote have a mitochondria, right? So chromoplast, any eukaryote have mitochondria, plus or minus chromoplast. Which ones have chromoplast? Plants and some algae known as the red algae. What's the function of the chromoplast? Photosynthesis. Do you remember the photosynthesis? What's the photosynthesis?
Sunlight and carbon dioxide, release glucose, oxygen, and ATP.
Good, but you take sunlight, water,
And carbon dioxide.
and carbon dioxide. Water and carbon dioxide. Is that clear? Which is very cheap, right? Plants do it from nothing. Like the water is everywhere. Carbon dioxide, you breathe it out. The plants take it, use the sun energy, so everything is free, right? And they make carbohydrates out of it. This is the function of the chromoplast. So can these green algae do photosynthesis?
Yes.
They can. Only green algae. Okay? They can do the same thing as the plants. They can make carbohydrates or sugar at the end. This is how it looks like. Okay? This is a chromoplast. But if you're talking about one of those, just one unit only, looks like coin, like this, they call that thylakoid. Thylakoid. Okay? A stack of those is called granium. Okay? Or granum. Granum. Thylakoid, granum. The whole thing is the chromoplast. Are we following? The chromoplast is like the mitochondria. There's a membrane from outside, and you have stacks of thylakoids. The stack is called granium or grana. Granium, which is one, or grana, which is plural. And there are two membranes. There are two membranes surrounding it, just like the mitochondria. Are we following? Remember this. Nucleus have two membranes, right? Double membrane. Mitochondria have a double membrane. Chromoplast, just like the mitochondria, it have a double membrane, inner and outer. Are we good so far? Any questions? We're fine? Okay. So next part is the cellular respiration. Cellular respiration is the function of the mitochondria. So the cells are respirating. Are you respirating?
Yes.
You are respirating, right? Right? What are you taking?
Oxygen, releasing
And what are you releasing?
carbon dioxide.
Your cells do the same thing. So we call it cellular respiration. The cells itself, like almost breathing, it's taking oxygen and releasing carbon dioxide, just like you. Okay? So if that's happening in the nucleus, in the cells, we call it cellular respiration. So you take oxygen and you use it to burn the sugar. Why are you burning the sugar? To get what out of it? To take energy and make ATP out of it. But what are you taking from burning the sugars? You're taking energy. you are going to store it in the form of ATP. Are we following or not? Is that okay? Did you get the idea? So here is the cell. I'm going to respirate, cellular respiration. I will take sugar in, and then I'm going to take some oxygen in. I use the oxygen to burn the sugar and make energy. Now I have extra energy. I should store it in the form of ATP. What's the ATP? What does it refer to? Abbreviation of what? ATP. Adenosine triphosphate. Adenosine triphosphate. It's adenosine and 3-phosphate. What tri means 3, right? Adenosine triphosphate. Adenosine and 3 phosphates. Okay? If this is, look at this. Let's say this is adenosine. My palm is adenosine, and these are the 3 phosphates. So this is adenosine triphosphate, right? Here is what's actually happening. If I need energy, I take off one of the phosphates and release energy. The energy is in between the phosphates. Did you get my idea? Adenosine triphosphate. This is the storage of energy. Where exactly do you store the energy? In between. So if I wanted to release this one, how can I release it? Remove one phosphate and release energy and use it. What do I have now?
2.
What do you call it? Adenosine. This was tri. What is this? Di. Now it's diphosphate, right? And you use the energy, right? Now I'm going to get some more sugar. I'm going to make some more, take some more oxygen, burn it, and get energy. I have extras. What should I do now? Put it in here and add phosphate again. I have it in the storage form. When you need it, break down on phosphate, release the energy, use it. When you get extras, put it back, put this back. So we always alternate between ATP, ADP, ATP, ADP. You need energy, break the phosphate, you have ADP, and take the energy. You have extras, reform the ATP again. Okay? This is the mitochondria, and the mitochondria have 2 membranes, outer and inner membranes, just like the chromoplast, just like the nucleus. They have 2 membranes, outer and inner. And the inner one is, look at this, this is folding. The inner one goes like this. Highly folded, unlike the outer. Outer is just a membrane like this. But the inner have a lot of folds like this. And there is fluid inside inside these inner membrane. This is where the cellular respiration is happening. And what's the cellular respiration?
Taking oxygen, release carbon dioxide.
You take oxygen and you release carbon dioxide. What are you going to use the oxygen for?
Sugar.
Burning the sugar. And then you end up producing carbon dioxide. Okay? We talked about the difference between plants and animals. What's in common between them, plants and animals?
Mitochondria and cellular.
What do you call both of them combined? Eukaryotes. Isn't plants and animals?
Yes.
Both of them are eukaryotes?
Yes.
Both of them are eukaryotes, right? Okay. So, being a eukaryote, there are things in common, right? Nucleus in common.
Cell membrane.
Cell membrane is, but cell membrane isn't even in the prokaryotes. What's in the eukaryotes? Nucleus, mitochondria. We call it the membranous organelles. In general, it is endoplasmic reticulum, golgi, apparatus. We just call it the membranous organelles. Can you remember that?
Yes.
You need to remember this. These are the basics. Otherwise, it's not a eukaryote. But number one is the nucleus with a membrane. Okay? Right. Now, since both of them, plants and animals, both of them are eukaryotes, but there are differences. What are the 3 differences?
Between the
Plants and animal. Both of them are eukaryotes, but what's the difference?
cell wall.
Cell wall in which one?
In the plant.
Yes, this is one.
Chloroplast in the plant.
Chloroplast in a plant.
The nucleolus inside the plant.
No, nucleolus is most of them.
Oh.
What's the third one?
The water sac.
What do you call it?
Vacuole.
Vacuole. It's a sac, but you call it vacuole. Vacuole. So did you get the 3 differences? I'm reviewing as we go, but you have to remember this. So here are the vacuoles. Do you have to have one vacuole only? No, you have to have one big vacuole. But you can, if you have extras, you can make smaller vacuoles. Is that clear? But you have to have a big one, which is the main one, the main storage. You can store here, you can store nutrients. Okay, you can store water if you have extras. You can store it temporarily here until you get it out. Pigments, anything can be stored in these vacuoles. And there is always one big vacuole, plus or minus others. But you have to have a big one in the first place. Some of the eukaryotes, which are the cells, or not necessarily eukaryotes, some cells in general, they can have a flagella or cilia. We talked about the flagella before. What is it? Tail. So what's the function of that tail?
To propel himself.
To movement. So flagella is for movement. The example here is the sperms. The sperms have tails. This is temporarily. The sperms have tails temporarily, and then when the sperm fertilizes an egg, the sperm is lost. The tail is lost. But anyway, normally it has a tail, so it can move. How about the cilia? Cilia is to swipe things around, to brush off things, looks like a brush. Okay, so you can brush things off. You have something like that in your trachea, for example. When you do when you do this, your cilia are pushing up secretions, right? Sometimes you do this and you might spit out, right? When you spit, the sputum, it was secretions inside and the cilia is pushing it up. So the cilia is to swipe or to brush off things out. It's tiny finger-like or hair-like projections that help to move things around, like these. This is not for the cell itself to move. The flagella is the cell itself move, like a sperm. But the cilia is not for that. The cilia cells doesn't move. You're just moving things around. Did you get the idea?
Yes.
So prokaryotes have a cell wall, and two of the eukaryotes have a cell wall as well. We talked about the plants before, right? Fungus is another one that also have a cell wall. That's why cell wall is not unique in prokaryotes. What's unique in prokaryotes? Capsule. You have capsule in the prokaryotes. You cannot have capsule in eukaryotes. Both of them have a membrane, yes, 100%. But prokaryotes have a wall. Can we have a wall in some of the eukaryotes? Yes, and that's why I'm saying the cell wall is not unique. Most commonly, it's more in the prokaryotes, but it can be, like plants and fungus. And in plants, the wall is rigid. The wall can be rigid because it's made of cellulose. Do you guys remember when we talked about the carbohydrates before? We did talk about it, right? Carbohydrates, fat, proteins, and we said carbohydrates can be monosaccharide, disaccharide, or polysaccharide. Do you remember this?
Mm-hmm.
We talked about it. What are the polysaccharides that you know?
Starch and cellulose.
Starch and cellulose. This is in plants or animals? Plants. What's the animal form of polysaccharide?
Cellulose.
No, cellulose in plants. Close, you're very close. Yes, glycogen. Polysaccharide in animals is called glycogen. Polysaccharides in plants is called
Cellulose.
Starch is number one, and cellulose is an additional one. Okay, there is one remaining that's more in insects. Chitin. This is the fourth one. Okay, but what is the edible one of all of this? Which one you can eat and neutralize out of the four? Starch. It's not glucose. Glycogen. Yes, glycogen. You can eat and neutralize. Starch from plants, glycogen from animals. The other two, no.
Yeah.
Why? We don't have enzymes to break it down. If you do that, like, well, what if I get some leaves from apple tree and eat it? Nothing. You're just going to break it down and it go to the feces as it is. Can I break it down and get some? No. Why? Because you do not have the enzyme. Animals can do that, right? They can eat that, certain things, and they can break it down. We cannot break down cellulose. So this cell wall, it can be rigid, and because it contains cellulose, and that's why plants can be upright like this. Like if you're wondering how the plant is like this, why it doesn't bend, how does grow like this? Because of the cellulose, a lot of cellulose, and cellulose is rigid, so that's why it's making it rigid like this. Next organelle is the cytoskeleton, and I talked about this before. Cyto means cell, skeleton, just a skeleton. So cytoskeleton is the skeleton of the cell. Like you have a skeleton, the cells have skeleton, right? So the cytoskeleton is the skeleton of the cells. Why do you have a skeleton of the cells? Same reason. Why do you have a skeleton? Why do I have a skeleton? So I can stand up, I can keep my shape, right? I don't collapse, I can move, right? When you move, aren't you using your bones and joints, right? So the same reason for the cytoskeleton in the cells. Now, if you take it to the next level, the animal cells. We talked about the animal cells, right? Here is an animal. Is the animal cell eukaryote or prokaryote? Eukaryote.
Yeah.
So here's an animal cell, another animal cell, another animal cell, another cell, another cell, another cell. What do you call these combined?
Extracurricular.
Somebody said it? Tissue. Do you remember this?
Yes.
We talked about atoms, molecule, organelles, cells, tissues. What's after tissues? Organs after organ system, and then organism at the end, right? So if I put these cells together, I call it tissue. But if you put it together, you have to put something in between the cells. So the cells stay together as tissue, and also they can communicate. And this thing in between is called extracellular fluid or extracellular matrix. It's that fluid in between the cells. Sometimes we call it interstitial fluid. This is holding them together, filling the space, and they can communicate through it. Otherwise, it is not tissues. It's individual cells if you don't have this. But we do have it. Okay? So that's it for this one here.